So I want you to try a little thought experiment with me. Imagine a tumor.
Usually not a very pleasant thing to imagine.
Right, no, definitely not. But usually we just picture it as this, chaotic lump of rogue human cells.
Yeah.
Just malfunctioning tissue.
Yeah, that's the standard textbook view.
Exactly. But today, we are completely shifting that perspective. I want you to imagine that tumor, not just as human tissue, but as a bustling alien city.
A city, I like it.
Yeah, a city that is just teeming with distinct microbial life. We are talking about bacteria, fungi, viruses, and even parasites.
Mm-hmm.
And they're all just setting up shop and thriving right inside the cancer.
It's a completely wild way to think about oncology. I mean, we're so used to viewing cancer as just this internal human failure, right? Not an ecological habitat.
Right.
But that habitat is exactly what we're going to unpack today on this deep dive.
And our roadmap for this whole journey is a frankly monumental 2026 paper. It was published in the journal Cell by Dolman and colleagues.
Oh, yeah. Biodiversity and Biogeography of the Multi-Kingdom Cancer Microbiome.
That's the one. And we are essentially embarking on a forensic investigation today. The researchers took this massive data set, over 16,000 tumor genomes.
Which is just a staggering amount of data.
It really is. And they used it to figure out exactly where microbes live in cancer, how they get there, and, frankly, why previous scientists got it so incredibly wrong.
Because they really did get it wrong. The scale of this study, it fundamentally rewrites the map. We aren't just, adding a few neat details to the literature here.
We're doing a teardown.
Exactly. We are tearing down a massive misconception in cancer research and building a totally new foundation from scratch.
Which honestly brings up a pretty glaring question for me. Mm-hmm. Scientists have been analyzing tumor DNA for decades, right? Using whole genome sequencing and all that.
Oh, yeah. Since the early 2000s, easily.
Right. So we've been staring at this stuff for a long time. Why are we only just now figuring out which bacteria are actually in there? Isn't finding bacterial DNA inside human DNA kind of like, finding a needle in a haystack?
Well, we actually need to heighten the stakes on that analogy a bit.
Okay, how so?
Because it's not just finding a microscopic needle in a haystack. It's trying to find that microscopic needle while someone is standing there actively throwing thousands of fake needles right at your face.
Wait, fake needles? Where are fake needles coming from in a highly controlled sterile genetics lab?
So to understand the fake needles, we have to look at the mechanical reality of whole genome sequencing, or WGS.
Okay, break that down for me.
Right. So when you biopsy a tumor, you grind it up and you run it through a sequencer. And what you get back is this massive digital file of A's, C's, T's, and G's.
Just millions of letters of genetic code.
Billions, actually. The sheer volume is staggering. But here's the catch. 99.9% of those DNA reads belong to the human host.
Because it's human tissue.
Exactly. So to find the microbes, you have to computationally subtract that entire human haystack.
So you basically run a filter, you tell the computer, hey, delete all the human stuff and show me what's left.
Precisely. But the problem, the really big problem, lies in the filter itself. For years, the global standard for that subtraction was an older reference genome called GRCH38.
GRCH38.
Kitschy. Right. But it was the best map of human DNA we had at the time. The issue is it was incomplete. It had massive gaps, especially in these really highly repetitive regions of our DNA.
Oh, I see where this is going.
Yeah. So because the computer didn't have a perfectly complete map of human DNA, it looked at these unmapped repetitive human fragments and just assumed they were alien.
Wow. So the human DNA basically slipped through the filter, put on a fake mustache, and disguised itself as microbial sequences.
That is exactly the origin of the fake needles. And previous pan-cancer studies relied on that imperfect filter.
Which means their results were just flooded with fake bugs.
Completely flooded. It led them to claim that microbes were everywhere. They published highly cited papers saying bacteria were naturally living in breast cancer, brain tumors, bone cancer, you name it.
That is wild. I was actually trying to picture how the researchers in this 2026 paper solved this, and I came up with this analogy. Tell me if this tracks.
Let's hear it.
Okay, so imagine you are trying to filter out the sound of a roaring symphony orchestra.
Right.
Just because you want to hear a single person whispering in the back row.
I like this.
Using that old reference genome was basically like having incomplete sheet music. You couldn't digitally mute the whole orchestra because you literally didn't know all the notes the brass section was playing. So those brass notes leaked through and sounded like whispers.
That is a perfect way to visualize it. And the new pipeline they developed in this paper, which they call PathSec-TTT, it fixes the sheet music.
Oh.
It uses a revolutionary new reference genome called the T2T-CHM13.
T2T meaning telomere to telomere.
Exactly, end to end. It is the complete, unbroken sheet music of human DNA. By cross-referencing this pristine, perfect map, the PathSec T2T pipeline can cleanly and completely subtract the roaring symphony.
And the researchers, they didn't even stop there, because I was looking at their methodology, and they are incredibly thorough. They actively filter against polymorphic human immune loci, known breakpoint junctions, and even vector sequences.
Yeah, let's break that down because it's super crucial. Our immune genes, those polymorphic loci, they are the most highly variable parts of our entire genetic code.
Because they have to constantly adapt to fight off different diseases, right?
Exactly. And because they vary so much from person to person, older reference genomes really struggle to recognize them. They just look like random noise.
So they got flagged as fake needles.
Right. But PathSec T2T specifically targets those hypervariable regions. It ensures no human immune DNA gets mislabeled as a bacterium ever again.
It basically scrubs the data clean. And then, just to be paranoid, they don't even rely on one tool to identify the whispers that are left over.
No, they use 3.
Right. They run the remaining data through 3 entirely different microbial classification tools. Kraken-2, Metaflan-4, and SILF. They just cross-validate everything.
And the benchmarking they did to prove this pipeline works, it borders on the absurd.
Oh, the computer simulation.
Yeah, the in silico stress test. They digitally mixed microbial DNA into human DNA down to a dilution ratio of 1 to 100,000.
One single microbial read for every 100 million human reads. I mean, that is an impossible signal-to-noise ratio.
It is. Out of over 300 million input reads, they buried exactly one single microbial read pair, just 2 tiny fragments of a bacterium called Veillonella rugosae.
And did PathSeq T2T find it?
It chewed through all 300 million reads, successfully deleted the human DNA, and accurately flagged that one single bacterial pair.
That is unbelievable. But okay, doing it in a computer simulation is one thing, right? The math is perfectly clean.
Right, the real world is messier.
Exactly. So they also ran an in vitro test. They mixed a standard microbial community into an actual petri dish with a real colorectal cancer cell line. I think it was called HCT116.
Yep, the HCT116 cell line.
And this is where the paper just takes a hilariously weird turn for me. Because during this real-world test, they sequenced the sample and they found cow DNA.
Yep, Bos taurus.
Cow DNA mixed right in with human colon cancer cells.
I have to ask.
How on earth does a cow wander into a sterile, high-tech genetics lab?
Well, it's all about how we keep human cells alive in a petri dish. They require nutrients to grow. And the global standard for feeding cultured cells is something called fetal bovine serum.
Bovine, cow.
Exactly. The researchers extracted DNA from the cancer cells, but the ambient DNA from the cow serum was so pervasive, it survived the entire sequencing process.
Wow.
It's actually a brilliant accidental proof of a terrifying reality in this field. Contamination is literally everywhere.
The reagents, the plastic kits, the air in the room.
All of it. So even if you have the perfect computational filter for human DNA, you still have to deal with physical, environmental smudges on your physical samples.
So how do you fix that?
To combat those smudges, the team developed a brilliant metric. They called it the Pan-Cancer Equiprevalence Score, or the PCE score.
Okay, if I'm tracking the math on this, the PCE score is basically a highly sophisticated BS detector for lab contamination, right?
That's a good way to put it.
It makes me think of a crime scene investigator. Like if a detective finds the exact same fingerprint at 20 totally unrelated murder scenes across the city, that person isn't a master serial killer.
Right.
It's just the detective's own fingerprint smudging the glass at every single scene.
That is the exact logic of the PCE score. It measures how evenly a microbial species is distributed across all the different cancer types in the data set.
Okay, so if it's everywhere, it's suspicious.
Highly suspicious. If a bacterium is showing up at equal, steady rates in a brain tumor, a lung tumor, and a colon tumor, it earns a high PCE score approaching 1.0.
And what was the cutoff?
The researchers established that any species with a PCE score greater than 0.7 was simply an environmental smudge.
So who were the biggest offenders? Like which bacteria were masquerading as the serial killers all these years?
The most prominent were really common skin-dwelling bacteria. Things like Cutibacterium acnes and Staphylococcus epidermidis.
Skin bugs.
Exactly. Along with ubiquitous environmental bugs like Pseudomonas aeruginosa, These are the exact microbes that flake off a researcher's skin or just live naturally in the lab water supply.
Wow.
And they effectively tricked a whole generation of scientists into thinking completely sterile tumors had massive microbiomes.
Okay, so just to take a quick pause for you listening at home, let's do a fast recap of where we are. We've seen how previous tumor studies were completely flooded with fake human DNA needles and lab contamination.
Right.
But now, PathSeq T2T uses a complete human genome to filter out the host, and the PCE score acts as a BS detector for lab contamination.
Exactly.
So just think about your own workspace right now. How easy do you think it would be for your own skin bacteria, like Staphylococcus epidermidis, to end up in a sterile lab sample? Keep that in mind as we start looking at the real survivors, because that brings up the ultimate question.
Which is,
once you delete the fake needles and you wipe the glass clean of those smudges, what does the true landscape of the cancer microbiome actually look like?
honestly, it looks incredibly sparse.
Really?
Yeah. Once the ultra-strict filtering was applied to all 16,000 tumors in the dataset, the vast majority of the microbiomes simply vanished.
Oh, wow.
Tumors in the brain, the breast, the skin, the lungs, they are essentially sterile. They lack any microbial community that is distinguishable from background noise.
So the earlier scientists were quite literally just chasing ghosts. For most cancers, there never was a needle in the haystack.
The paradigm shifts completely here. However, the study did identify one massive, undeniable exception.
Okay, what's the exception?
Cancers of the orodigestive tract.
Meaning everything from the mouth down through the stomach and the intestines.
Yes. Oral, esophageal, gastric, and colorectal cancers. These tumors consistently harbor robust, highly complex microbial communities that actually survive all the decontamination filters.
Okay, I suppose that makes anatomical sense, right? Because the digestive tract is basically an open highway running through the center of our bodies.
Yeah, that's a great way to look at it.
Food, liquids, and millions of microbes are constantly passing through there anyway. So a tumor growing on that highway is basically like a biological rest stop where these microbes can just pull over and park.
Exactly.
But surely these rest stops aren't just, holding tanks for bacteria. There has to be more going on.
Well, you're touching on one of the most significant revelations in the entire paper. It is not just bacteria.
What else is there?
By expanding their search parameters with this new pipeline, the researchers proved these tumors are actual multi-kingdom ecosystems. They found representatives from across the entire biological spectrum.
Okay, let's catalog this alien city then. What else is setting up shop in these tumors besides bacteria?
Well, we could start with fungi.
Fungi, like yeast.
Exactly. They found Candida albicans highly enriched in esophageal and gastric cancers. And even more surprising, they found Saccharomyces cerevisiae.
Wait, Saccharomyces cerevisiae, isn't that literally baker's yeast?
It is the exact same stuff used to make bread and beer. And they found it actively colonizing the lower part of the stomach, specifically the gastric antrum and pylorus, as well as the lower gastrointestinal tract.
Baker's yeast thriving inside a stomach tumor. That is wild. Okay, what else is in the city?
Archaea.
Oh, the extremophiles.
Right. These are ancient, single-celled organisms that are entirely distinct from bacteria. You usually find them in deep-sea vents or hot springs.
But they're in human tumors.
Yep. The data showed Methanobrevibacter smithii and Candidatus methanomycelicoccus intestinalis.
That is a mouthful.
It is. But basically, these are methane-producing archaea, and they were localized specifically to tumors in the lower GI tract.
Why only the lower GI tract?
Likely because they are strict anaerobes. They require an environment with absolutely zero oxygen, which the lower gut provides perfectly.
Okay, so wait. You've mentioned bacteria, yeast, and archaea. What about viruses? I mean, I know certain viruses like HPV are known to cause cervical cancer, but do viruses actually take up residence inside these digestive tumors too?
They absolutely do. Human papillomavirus, specifically the HPV-16 strain, was uniquely dominant in oropharyngeal cancer.
Up in the throat.
Right. They also tracked Epstein-Barr virus, making significant appearances. And perhaps the most shocking finding of all, they detected rare instances of protozoa.
Protozoa, wait, like actual swimming parasites.
Specifically, the parasite Trichomonas.
Oh, wow.
So when we call it a bustling alien city, I mean, it is not an exaggeration in the slightest.
That is terrifying and fascinating. And what really gets me is that this multi-kingdom city isn't just a random assortment of squatters, right? Like the researchers mapped out what they call the biogeography of the tumors.
Yes, the biogeography is key.
The microbes reflect the exact anatomical zip code of the tumor.
Because the geography dictates the ecology. For example, proximal colon cancers. Those on the right side of the colon, they have a completely different microbial profile compared to distal or left-sided colon cancers.
Just based on which side of the colon they're on.
Exactly. And up in the mouth, oropharyngeal cancers harbor periodontal pathogens like Porphyromonas gingivalis.
Gum disease bacteria.
Right, but then down in the stomach, gastric tumors act as a haven for lactobacillus.
Which makes sense because lactobacillus is used to make yogurt, right? It loves acid.
Spot on. It makes evolutionary sense because those bacteria are heavily acid tolerant. They can survive the stomach acid that would kill the gum bacteria.
So even when human tissue mutates and becomes cancerous, the local environment still acts as a bouncer. It decides who gets into the club based on the rules of that specific organ.
That's a great analogy. It suggests that tumors don't just invent a new microbiome from scratch. They recruit from the local mucosal population that is already adapted to survive in that specific pH and biological neighborhood.
Okay, so let's do another quick recap for you guys listening. We are at the 10-minute mark here. We've gone from the old idea that every tumor is crawling with bugs to the reality that most solid tumors do not have a microbiome at all.
Right, they're mostly sterile.
Except for orodigestive tumors, which are rich ecosystems containing bacteria, fungi, archaea, and viruses. And these microbes reflect the exact anatomical zip code of the tumor.
Perfectly summarized.
So ask yourself, if a tumor in your colon can harbor entirely different fungi and archaea depending on which side of the colon it's on, what does that tell you about how highly specialized these microenvironments actually are?
It shows how precise the ecology is.
Right. But honestly, an accurate map just introduces a totally new mystery for me.
How so?
Well, if we zoom in on a single zip code, let's just say the proximal colon, you can have two tumors right next to each other on the exact same stretch of highway.
Okay.
One tumor has a massive bacterial load, maybe 30 bacteria for every 1,000 human cells. But the tumor right next door, literally an inch away, has almost none.
Yes, that is a real phenomenon.
So why is one rest stop packed with cars while the other one is completely empty? They are in the exact same environment.
Because the answer isn't in the environment. It is hidden inside the tumor's own broken DNA.
Wait, how does the DNA of the human tumor affect the bacteria?
So the researchers analyzed hypermutated genomic subtypes of cancer. They focused on tumors with microsatellite instability known as MSI and polymerase mutations, or POL.
Okay, those are big genetic terms.
Yeah, but simply put, in these specific tumors, the fundamental mechanisms that repair DNA are totally shattered.
Okay, let's visualize that. So if a healthy cell's DNA repair process is like a really good spell checker on your computer, constantly fixing genetic typos as they happen.
Right.
A hypermutated tumor is basically a factory with a completely broken spell checker, just aggressively pumping out millions of genetic typos.
That is exactly what is happening. And that sheer volume of typos is measured as tumor mutation burden, or TMB.
TMB, got it.
And the mathematical correlation the team discovered regarding TMB is arguably the centerpiece of the entire paper. They found a strict linear relationship between the number of genetic typos in the human tumor and the sheer volume of microbes living inside it.
Really? Give me the math. How strong is the link?
It's incredibly strong. For every 10-fold increase in tumor mutation burden, there is an average 4.7-fold increase in microbial abundance.
Wait, a 4.7-fold increase. That is a staggering jump.
It is massive.
And that holds true across the board.
It is incredibly robust. The correlation held across multiple genetic subtypes, and they even validated it across completely independent data sets.
Oh, to make sure it wasn't a fluke.
Right. They compared the 100,000 Genomes Project in the UK against the Cancer Genome Atlas in the US. The results were identical. The broken spell checker literally acts as a magnet for microbes.
But which microbes are answering the call? Like, who actually wants to live in a highly mutated tumor?
The primary colonizers are specific species that actively seek out these high TMB environments. The most infamous one is Fusobacterium nucleatum.
Okay, Fusobacterium.
Alongside a related subclade called Fusobacterium animalis and another aggressive species named Selenomonas sputigena.
Okay, but I am genuinely struggling to understand the why here.
The mechanism?
Yeah. Why does a bacterium care if the human tumor cell has a lot of DNA typos? A bacterium doesn't read human genetics. How does it even know the tumor is highly mutated in the first place?
That is the secret. It doesn't read the DNA. It reads the immune system's reaction to the DNA.
Wait, what?
This is the most elegant mechanism in the entire study. It's called immune exhaustion.
Okay, walk me through this.
So when a tumor has a massive mutation burden, all those genetic typos cause the cancer cell to manufacture weird, deformed proteins on its surface. We call these neoantigens.
And the human immune system is designed to hunt down and destroy weird proteins.
Fiercely, T cells spot these neoantigens, recognize them as a major threat, and they launch a massive coordinated attack.
Which means lots of inflammation.
Exactly. This creates a huge, localized spike in inflammation around the tumor. But here's the critical failure point. The tumor doesn't just die.
Because it's cancer. It keeps growing.
Right. It keeps pumping out weird proteins, and the immune system keeps attacking over and over until the immune cells literally burn themselves out.
Oh, wow. They get exhausted.
They experience profound immune exhaustion. The T cells begin expressing immune checkpoints, which are essentially biological off switches. The local immune system just stops fighting and shuts down.
And that creates a bubble.
Yes. It creates a localized, completely immunosuppressed bubble surrounding the highly mutated tumor.
It's a safe house.
Exactly.
The tumor accidentally builds a fortress. By causing so much genetic chaos, it exhausts the immune system, creating a dead zone where bacteria like Fusobacterium can just move in, set up camp, and multiply rapidly without any white blood cells coming to fight them off.
The tumor's genetic chaos actively re-engineers the local ecology. The cancer creates the void, and the microbiome fills it.
That completely flips how I view cancer. So let's just recap this really quick for our listener. Tumors with highly mutated DNA, high TMB, attract drastically more microbes. Specific bacteria like Fusobacterium actively seek them out.
Yep.
And the theory is that the mutated tumor exhausts the local immune system, rolling out the red carpet for these microbial invaders.
That's the mechanism.
So, for you listening, does it change how you view cancer? Knowing that the tumor's genetic chaos isn't just affecting the human cells, but is actively reshaping the local ecosystem to invite bacteria in. It's wild.
Yeah.
But the paper doesn't just stop at the genetics of the tumor, does it?
No, it doesn't. They do a pretty major pivot.
Right. They pivot from the genetics of the cancer to the demographics of the patient, specifically their age.
Yes. And this brings us to a, frankly, terrifying trend in modern oncology. The global rise of early-onset colorectal cancer, meaning patients diagnosed under the age of 50.
It's one of the biggest mysteries in medicine right now. Everyone is talking about it. How did they investigate it using this new data?
They isolated a highly specific subset of the data. They looked at distal tumors, meaning cancers on the left side of the colon that were microsatellite stable.
Wait, stable, meaning these tumors did not have that broken spell checker.
Correct. Their mutation burden was very low.
Okay.
Then they compared the microbiomes of the patients under 50 against the patients over 50 within that specific low mutation group.
Let me guess. The young patients had a totally different alien city.
Right.
A bunch of crazy new bugs.
Actually, no, it wasn't about what was there. It was about what was uniquely missing.
Missing.
Yes. In the young patients, one very specific bacterium was profoundly depleted. It was almost gone. It's called Akkermansia mucinifila.
Akkermansia mucinifila. Okay, let's unpack that. What is its day job in a healthy person, and why is it so catastrophic that it goes missing in young people?
So Akkermansia is a commensal bacterium, meaning it naturally lives within our gut and provides a mutual benefit.
Okay, it's one of the good guys.
Right. And its specific biological niche is mucin degrading. It survives by literally eating the protective mucus layer that lines the inside of your intestinal walls.
Wait, hold on. That sounds like friendly fire. Why would we want a bacterium actively eating away our protective mucus shield?
I know, the mechanism is wonderfully counterintuitive, but by constantly nibbling on the mucus layer, Akkermansia acts as a localized stressor.
A stressor.
Yeah. This mild constant stress stimulates the host's epithelial cells to regenerate and produce even more mucin. It literally forces the human body to build a thicker, healthier, far more robust biological barrier.
Ah. That's like a microscopic drill sergeant for your gut barrier. It stresses the wall just enough to make your body build it back stronger and thicker.
Exactly. It is the biological architect of that space. And because of this continuous reinforcement, robust populations of Akkermansia are heavily linked to metabolic health, longevity, and drastically reduced intestinal inflammation.
But in the young patients developing early-onset colon cancer, the drill sergeant has gone AWOL. The Akkermansia is missing.
And the mechanical cascade of that absence is severe. Without Akkermansia constantly stimulating the barrier, that protective mucin layer can become dangerously thin, sluggish, or totally dysfunctional.
And when the wall weakens.
Exactly. When the wall weakens, luminal genotoxins, which are toxic, DNA-damaging chemicals naturally passing through your digestive tract from your diet or the environment, they can easily penetrate that weak mucosal shield.
Oh, wow.
And they make direct contact with the unprotected intestinal tissue.
So the wall falls into disrepair, the dietary toxins leak through, they physically damage the DNA of the colon cells, and they spark an early-onset cancer.
That's the leading hypothesis, yes.
That is a chilling but incredibly clear chain of events.
It really highlights how precarious the balance of this ecosystem truly is. A single missing species of bacteria can compromise the host's physical armor, leading to a cascade that ends in a malignancy.
Man, this whole journey today, it changes everything about how we need to approach this disease. I mean, we started by watching the researchers use the ultimate complete human genome to debunk the ghost bacteria, wiping away the environmental smudges to reveal the true map.
And that precision is not just an academic exercise. We cannot treat cancer effectively if we are building drugs to chase ghosts.
Absolutely. But once the map was clear, we saw that these orodigestive tumors are vibrant, multi-kingdom ecosystems. Just a complex alien city of bacteria, fungi, archaea, and viruses, all strictly organized by their anatomical zip code.
Beautifully put.
We learned the rules of the city, too. How a broken DNA spell checker exhausts the immune system to build a safe house for fusobacterium. And finally, we saw the tragic cost of a missing biological architect, like Akkermansia.
Which brings us to the real hope here. Now that we know the mechanisms of exactly who is living in these tumors and exactly why they are there, the medical field has entirely new, highly specific targets for intervention.
Which leaves me with the final, slightly provocative thought for you to take away. For everyone listening, if these orodigestive tumors are not just lumps of human cells, but complex ecological habitats relying on a specific balance of fungi, bacteria, and immune exhaustion, could the future of oncology look less like traditional medicine and more like wildlife conservation?
Wow, it requires a total paradigm shift, moving from a mindset of sheer eradication to a mindset of ecological management.
Exactly, because instead of just flooding the human body with maximum toxicity poison, hoping to kill the tumor before it kills the patient, what if the oncologists of the future act as park rangers?
I love that.
What if they introduce targeted viral phages, the viruses designed specifically to hunt down the fusobacterium hiding inside those immune safe houses? Or what if we start prescribing personalized Akkermansia probiotics to young people to rebuild their biological walls before the cancer even has a chance to spark?
Treating the environment to heal the host, it really is the frontier of precision medicine.
It really is. So the next time you hear the word microbiome, I want you to remember that alien city. Remember that a tumor is a living, breathing ecosystem. And maybe learning how to manage that city is the key to finally tearing it down. Thank you so much for coming on this deep dive with us today. We'll catch you on the next one.